Hi, and welcome to the regulation of eukaryotic gene expression. Exactly. Control of gene expression in the eukes. That's what we're talking about. More complex than in the prokes, just like everything else. Differential gene expression. What does that mean? Different genes turned on and off in different cells. On or off. Different cells. People. And there are 5 points for control of gene expression. 2, the only ones that happen in the eukes. This one and this one, right? Not prokes. Just the eukes. And so what are these 5 points of control? Chromatin remodeling. Ooh, transcription, yes. Processing, yes. Lifespan of the mRNA, right? Remember, that's controlled by the tail, poly A tail. Remember that guy? Poly A tail. Woohoo. Long tails. Takes a long time to eat those guys off. And what else? Translation. Post-translational control. And there we have it. They don't really talk about alternative splicing, but we'll get to that. So don't worry. Never fear. We will get to that. It's going to be fine. All right, packaging and regulation. This doesn't happen in bacteria, right? Remember, that's just our little circle guy, right? Our bacterial chromosome is just one circle. But Euk DNA, right, is linear double helix, right? And usually it's the spaghetti shape, right? We should draw it like that. It's not in its chromosome sausages, right? When do chromosome sausages happen? Prophase or prophase 1. And so normally it's a spaghetti shape, but that doesn't mean it's just naked. That doesn't mean it's just this double helix. Okay, so we're going to talk about that. Let's move on. Chromatin structure. Yay. It contains nucleosomes. They are bead-like structures. Of proteins, histone proteins that the DNA wraps around. Okay, so if you look at it under a microscope, it looks like beads on a string. So each one of those little guys is the nucleosome. There's DNA wrapped around it. Here's our nucleosome protein. There's our DNA wrapped around it. And then there's the linker region, another histone complex, DNA wrapped around it, another linker region. So the little straight guys, linker, there's nucleosome, linker, nucleosome, linker, nucleosome. Let's look at a picture, shall we? I think we shall. And so here's the same picture, except it's a lot better than mine. So here's, right, it's actually 8 proteins, an octamer, ooh, and then another little H1 histone in there to help the linker guys. The DNA's wrapped around. Here's that string, bead, string, bead, string, beads, string, and then it's usually wrapped up about like that. Okay, and so this is normal. This is chromatin, right? So this is open chromatin, regular chromatin, not chromosome. So even in chromatin, which is spaghetti, right? When we draw the spaghetti, it still has some structure with those proteins, okay? Okay, yes, you're okay. More like this, we might call that closed, and we might call this open. And that's still chromatin, still spaghetti. It's even more open in active genes, so it's a little bit more closed, right? So a little bit, but the regions where transcription can happen, where genes turn on, right? It has to be a little more open. Otherwise, we compact them down so we don't transcribe it, or we get very little. What do I mean by it? If it's condensed like that, it's going to have no transcription. If it's open, decondensed, open, right? We're going to do yes transcription. Histone deacetylases, histone acetyltransferases. We will talk about those guys. Kind of. A little bit. You just really need to know condensed chromatin, right? We call that heterochromatin. Chroma. I ran out of space. Heterochromatin and euchromatin. So many terms. What the heck? All right. Okay. So in euchromatin, we still have to have something that gets us to the promoter, right? And allows us to start transcription. We talked a little bit about this in the regular transcription unit.
Bruh.
Yeah, I know. I'm going to tell you again, right? You're just going to figure it out. So remember, this is our double-stranded DNA. Here, boink, 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 boink. And this was our +1 of transcription, right? +1 of TSS. It depends on who you talk to. We showed you that the core promoter was just up here, but, you know, in the real world, it can be down here also. We're just going to talk about up here. Remember, there's that minus about to -40, -50. What's at the -30 in our classic genes? Tata box. Right? That's where TBP binds and then brings in RNA polymerase. RNA polymerase 2. Right? And then we get transcription to go. So that's the core promoter, what binds the TBP, RNA polymerase, and all those other friends. There's also more promoter up here in the DNA, right? Remember, the promoter is double-stranded DNA. It's a sequence. So this is a proximal promoter. That's the part that I love. I love me some promoters. I can't help it. They're like the best things ever. Interesting. Okay, so most regulatory sequences, parts of DNA that control the gene, right? Upstream of the core promoter, upstream in the negative, so not -500 base pairs. Whoa, crazy. Okay, so promoter proximal, again, core promoter, we would say tataboxy guys in their start site, transcription start site, right? That's where go exon, intron, right? Somewhere in here we'd have an ATG, right? Somewhere in here we'd have a stop, but not at the very end, right? ... Those guys are going to bind here. They help TBP and RNA polymerase do transcription. Go, faster and faster. Give me more transcripts. What else is there? There's also things called enhancers. Again, sequences that can be way far upstream or downstream, and they can bend around and help the promoter proximal elements. They can help the core promoter. Again, really turn up transcription. Let's take a look at a picture. Oh, let's not take a look at a picture. What the? Wow. Okay. Easy. Oh boy. Okay. Stop it. Okay, silencer elements are similar to enhancers. They can be anywhere, but they can actually help repress genes. So enhancers enhance, right? Enhance, turn it up. Silencers silence or regulate it down. So modulating transcription. Not really on, off, but up, down. Yeah. Okay, what else? So here we go. Ooh. So what does this look like? You can, again, here's our enhancer. We can have one upstream, 10,000 base pairs away, 100,000 base pairs away. It can be downstream. It can actually be in an intron. It can be in another gene. Has to be on the same chromosome, though. Right, here's core promoter. Here's proximal promoter. Our start site. Woohoo! Exon, intron. right? When we do transcription, remember the start site here. This is where we would start. Oh, whoops. This is where we would start and we would end at the exon, right? 5' to 3', right? That's our RNA. And then we're going to take out 2 introns, 3', 5', right? So the exon's only here, right? There's our processing. Don't forget your cap. Cap it. Put the little hat on that guy, right? The upside down G methylated cap. And then don't forget our tail, to give us our mRNA. And then, boom, go get translated. Yeah, that's what we're talking about. How do enhancers work? Oh, well, they turn things up. We're not going to go through this experiment. Don't worry about it. We got enough to think about, enough to worry about. Transcription initiation, we kind of already talked about that, right? Basal transcription factors, core promoter, TBP, binds TATA sequence, brings in RNA polymerase, and then the regulatory guys can make this happen more, right? And so here we are talking about a little bit of a model, and here's all the good stuff. They're showing the enhancers, right? Because, right, it's DNA. It's linear, but it doesn't mean it can't bend. So how could an enhancer way out here do anything way down here? Well, it bends around. It could bend around. It could bend around, right? Sort of banging on RNA polymerase to go, go, go. Right? That's all it is. Transcription. Easy peasy. Again, what are the 2 unique things? Chromatin remodeling, heterochromatin, euchromatin, RNA processing. That's the best things. What else can happen? Post-transcriptional control, right? Primary transcript, we have to process it, but we could splice it in different ways. We could alter the rate of that starting translation. We can modify mRNAs. How do we do that? Poly-A tail length. Woo, poly-A tail. And then regulating that protein by stability after translation. I referred to that a little bit in class for translation. And we're just going to talk a little bit, like I mentioned, alternate splicing, because I don't want you to leave 107 and not know that that actually happens, right? So we always remove introns, and generally we keep the exons, but we don't have to keep all of them. Ooh, that's called alternative splicing. So let's take a look, shall we? I think we shall.
What the dog doing?
I don't know. Alternatively splicing some mRNAs, I guess. So here's our example. Bunch of exons, bunch of introns. And, right, we would do, I guess I don't know where the, I don't know. Is this our 3' UTR? We'll pretend. 3' UTR. Ooh. This maybe is our 5' UTR. We'll pretend like, no, I guess not. I guess that's another exon. Ooh. By looking at the picture. Ow, dang it. Then, okay, start again. Ignore that. Go. I know, it's annoying, right? Like, hello? Sorry, okay. I could redo this, but,
Bruh.
I know, I'm tired. Okay, all of these, I don't know why they didn't point to them. That's an exon. Right, the introns are in between exon. They're pointing to the colors. I guess I could have looked. And so they're saying when we do splicing, yeah, we got rid of all the introns, all of those guys. But in the case of skeletal muscle, right, both include this one. That's this. But then in the skeletal muscle, when we get rid of, let's cut it out, we get rid of that intron, we actually get rid of that exon as well. And so we're saying that guy gets connected right there to that guy. So we remove 2 introns and an exon. Then we remove that intron, all these guys, like regular people. All the exons go into there. And then we remove this one. These 2 purple guys, exons come in here. And then we splice the rest of this out and it leaves. We don't use it. In the smooth muscle, it's a similar protein, but not exactly. So again, that first part sticks in here, right? There we go, right there. But now, instead of splicing out the blue part, we keep that in. So we do a splice right here. And we put, right, that blue guy stays. But we don't want that purple guy. So we go from here to here. And then all of these, just like before, go into here. And then they're also saying we get rid of all of this. Right? That gets all spliced out. Gone. Poof. Poof, gone. And then that guy comes here. That's alternate splicing. Introns always come out. Some exons come out. Some exons stay in. I know, it's a little bit crazy.
Bruh.
Yeah, I know, but it's okay. We're going to be fine. Cell type specific proteins that regulate the spliceosome. So that's just the complex that does all splicing. Right? Lots of proteins in there. We don't want to learn what they are, but a complex of proteins that do the splicing, whether it's regular or alternative, same, right? It's the same thing. Yeah, I know, it's crazy. At least 70% of the human genes undergo alternative splicing. We thought, right, very few did, but they actually do. So we can make multiple proteins from the same gene through alternative splicing. Woo, that's awesome. All right, so then translational control. I promised I wouldn't talk too much about this. But, you know, I'm going to talk a little bit. It is very, very exciting, right? Okay, so we talked about stability of mRNA. That was our tail. Shorter tails, they survive longer. Longer tails, they survive a while. There's also some proteins that can bind mRNAs to block translation. Also, other RNAs. Ooh, can do that too by complementary base pairing. Woo! And then we can also slow or stop translation by phosphorylating some of the ribosome. So we mess with the ribosome, and that we can control a little bit of translation. Post-translation, I'm not going to talk too much about this. There's ways of dealing with proteins, making them active or not by phosphorylating. Right, it's much more complicated. Ooh, there's some ATP. Yikes, we have it inactive. Ooh, we put some stuff on it and it becomes activated. Then it can go in and do its job. Yay! We're not going to test you over that. But you know that it exists as a way of controlling how proteins work. Post-translational control. And then how do we compare all of this stuff? Well... Oh my god. Oh, what the heck? Sorry. Wait. Oh. Yeah, what the heck? Okay, so packaging accessibility of DNA, that's mostly Euks. Processing of transcripts, only Euks. Complexity of transcriptional control, right? Euks. More complex, so they claim. Coordinated expression, I don't know what they mean. If they mean operons, right? Where we get multiple genes transcribed together and then we translate them, right? We need one, we need all three. Or do they mean some sort of coordinated expression like tissue specific, right? And that would be tissue specific, like we talked about alternative splicing. So it could be either of those. But that's it, my friends. That's all I have. I'm sorry I couldn't give you more, but that's it. That's all I got. Thanks so much for watching.